Hello, my name is Luca and I'm a master's student from Acadia University. Today I'm presenting a poster on my master's research with the title Effects of imidacloprid for hemlock-woolly adelgid control on non-target pollinator communities, authored by Dr. John Sweeney, Dr. Matthew Smith, Dr. Jess, Dr. Kirk and myself. Hemlock-woolly adelgid is a destructive invasive forest pest that was introduced to eastern North America in the 1950s. As the name suggests, hemlock-woolly adelgid attacks hemlock trees and causes high mortality within a stand in just a few years. As part of an IPM plan, multiple different treatment methods are used to contain and manage the spread of this pest, including silviculture, biocontrol, and chemical control. Chemical control with pesticides is proven to be highly effective, and the injection of hemlock trees with imidacloprid, a neonicotinoid insecticide, is widely used in the US and Canada to treat infested trees. Unfortunately, treatment of a tree has to be repeated every 5 years as the protection wears off, and injection is labor-intensive and expensive. In the US, another treatment option, imidacloprid basal bug sprays, has shown promising results in managing hemlock-woolly adelgid infestation, and application is much quicker and cheaper. Thus, a greater number of trees could be treated with similar effort. This is where my project comes in. Even though imidacloprid basal bug sprays may present a promising alternative to tree injections, questions remain regarding its non-target effects on other arthropods. Imidacloprid is commonly used in agroecosystems and has been associated to play a role in the global pollinator decline. Even small sublethal doses of imidacloprid have shown to have detrimental effects on bees, such as reduced nesting activity, navigation memory, and foraging performance. In my project, I'm aiming to answer if the application of imidacloprid basal bug sprays in eastern hemlock forests negatively affect pollinator communities. We are investigating potential changes and declines in communities of two pollinator groups, bees and flower flies, following imidacloprid basal bug sprays in eastern hemlock forests at Mi'kma'ki, Nova Scotia. I would now like to talk about the general setup of the project as well as my methods. The study takes place in southwestern Nova Scotia at 4 different sites. On the map on the right, you can see the location of each site as well as the current spread of HWA in our province. All sites are dominated by eastern hemlock and infested with hemlock-woolly adelgid. They are also all subdivided into 4 circular plots of 36-meter diameter where experiments took place. Plots at McKay Lakes and Sissibu Falls were treated with basal bug sprays in fall 2020 and 2021. At Hemlocks and Heartwoods, trees were treated in 2022. Mill Falls remained an untreated control site for the duration of the project. Plots were treated with a maximum of 1.89 liters of the product Zytec 2F, which is an equivalent of 450 grams of imidacloprid per hectare per year. To determine whether pollinator communities underwent changes, we sampled all plots using multiple pollinator sampling techniques, such as blue vein traps, pan traps, and sweep netting over 2 years in 2021 and 2022. Surveying took place during the major flight period of pollinators from early May to mid-September. Trap specimens were collected bi-weekly. We plan to compare the diversity, abundance, and species assemblages between treated and untreated stands and between sample years in treated and untreated controls. Furthermore, we also wanted to determine the levels of imidacloprid pollinators may be exposed to within treated plots. During biweekly collection of trap specimens, we also collected pollen and plant material from flowers growing within treated plots. Pollen was also collected directly off of bees that were sweep netted. These samples will be analyzed for levels of imidacloprid and its metabolites to determine whether safe levels are exceeded. Lastly, we wanted to highlight plant-pollinator relationships within the sites. We conducted monthly ground vegetation surveys and recorded blooming periods of flowers on a bi-weekly basis. We are planning to make a temporal and spatial analysis of the relationships between pollinators, flowers, flowering plants, and blooming periods. Next, I would like to present some preliminary results of the project. We collected a total of 1,479 insect pollinators, 548 bees, and 931 flower flies over 2 field seasons in 2021 and 2022. We captured bees from 11 genera and flower flies from at least 19 genera, but identification is still ongoing. Bumblebees were the most abundant pollinators at all sites and made up a total of 78% of bees captured. In the first 4 bar graphs, we are showing the total number of total number of bees in yellow, and flower flies green at the 4 sites between 2021 and 2022. Each graph represents a different site. Graph A is Mill Falls, the untreated control site. Graph B is Hemlocks and Heartwoods, which was treated in spring 2022. C is McKay Lakes, and D is Sissebu Falls, which were both treated in 2021 and again in fall 2022. Generally, the number of bees went up in 2022 versus 2021 at all sites. Numbers of flower flies rose at McKay Lakes and Sissebu Falls, but they did decline at Mill Falls and Hemlocks and Heartwoods in 2022. In the next 2 bar graphs, we show the total number of Bombus tenarius in the left graph and Bombus terricola in the right graph, captured at Mill Falls and Hemlocks and Heartwoods in 2021 and 2022. Please keep in mind here that Hemlocks and Heartwoods was treated before the 2022 field season, whereas Mill Falls remained untreated. The number of Bombus tenarius increased at both sites in 2022 versus 2021, but Bombus terricola was absent from Hemlocks and Heartwoods in 2022 after it was treated. In the last 2 graphs at the bottom, we show the total number of bees and bee genera on the left graph and flower flies on the right graph captured per trap type. BVT stands for blue vein trap, SN for sweet netting, and YP, WP, and BP for yellow, white, and blue pan traps respectively. You can see that blue vein traps captured most individuals of bees as well as flower flies. Blue vein traps also captured all 11 bee genera encountered during the project. Next, I would like to talk about some preliminary conclusions and the next steps of the project. We did not find consistent differences in pollinator abundance between the imidacloprid treated plots and untreated controls in 2021, nor between pre versus post-treatment at Hemlocks and Heartwoods. Although no differences in overall abundance pre and post treatments were apparent, some species may be more susceptible to treatment than others. For example, Bombus terricola was not found post-treatment at Hemlocks and Heartwoods in 2022. Please keep in mind, however, that all of these data and conclusions are preliminary, and some of these conclusions may change after data analyses are complete. In the comparison of the trap types, it became clear that blue vein traps presented the most effective way to sample bees in eastern hemlock forests, as they captured most individuals as well as all 11 bee genera found in the project. Preliminary identification shows that blue vein traps are likely the superior way to sample flower flies as well. The next steps for the project are further ID of specimens to species level, including DNA barcoding of cryptic species to statistically compare diversity and richness between sites and years. Secondly, also the analysis of pollen and plant samples for imidacloprid levels. And thirdly, determining the relationships between pollinator richness and abundance and the presence of flowering plants, blooming periods, and canopy cover. Lastly, we would like to thank the individuals that contributed to this project, including Corey Hughes, Christopher Edge, Dave Ireland, Gavin Martin-Skapter, Brian Lorraise, Sally Steele, and many others. Special thanks also to Cody Chapman, Jago Camolet, and Taylor Swanberg for providing assistance with fieldwork. Financial support for the project was provided by the Canadian Forest Service, Parks Canada, Search International, and the Bloomington Naturalist Society. Thank you very much.